current challenges and new approaches to developing our AAV manufacturing process for large-scale GMP production. We are delighted to have you join us as we explore and innovate solutions to one of the most pressing challenges in gene therapy. AAV production faces significant hurdles, including low yields, scalable issues, and inconsistency in qualities. To meet the growing clinical and commercial demands, it's necessary to develop advanced and efficient manufacturing process. Today, we will introduce you to PackGene's proprietary Pi Alpha 293 AAV High-yield Platform, which addresses these challenges head-on. This platform employs a uniquely designed RC plasmid within the triple-plasmid transfection system, boosting AAV production by 3 to 8 times across various serotypes. We are honored to have two distinguished speakers from PackGene Biotech to join us today. The first one will be Dr. Berenice Carrillo, Associate Director of Process and Analytical Development. Dr. Carrillo oversees the design and development of robust, efficient, and scalable downstream processes using PackGene's proprietary AAV platform technology. She brings a wealth of experience from her previous role as Director of Downstream Process Development at iBio-CDMO. Dr. Carrillo holds a Bachelor of Science degree in biochemistry from New Mexico State University and a PhD in molecular biology and microbiology from Baylor College of Medicine. Her 20-year career as a protein biochemist includes significant contribution to protein engineering, purification, and crystallology. The second one will be Dr. Ren Li, Associate Director of Process and Analytical Development. Dr. Li leads a development team of AAV viral vectors for gene therapies at PackGene. His extensive background includes senior roles at CTMC and Bellicum Pharmaceuticals, specializes in viral vector production and T-cell therapies, as well as a senior scientist position at Close Pharma. Dr. Li holds a PhD in pharmacology and cancer biology from Duke University, with postdoctoral research experience at UT Southwestern and Texas A&M, and an undergrad degree from Tsinghua University. Before we begin, here's a few housekeeping notes. Please use the Q&A feature and submit your question throughout the presentation. Just type your question in the box and we'll be able to see them and answer them at the dedicated Q&A session at the end. This webinar is being recorded and will be available on demand for viewing after the session. Now, without further ado, let's hand it over to Dr. Li and Dr. Carrillo. Thank you.
Welcome, everyone. Thank you for joining us today for this webinar. I am Berenice Carrillo, Associate Director of Downstream Process Development, and presenting along with me is Ran Li, Associate Director of Upstream Process Development. Today in this webinar, we are going to discuss the current challenges and new approaches to developing AAV manufacturing processes for large-scale GMP production. In this webinar, there are 5 main points that we will be presenting. First, we will give an overview about the current challenges and the status of AAV-based gene therapy. Next, we will give you an overview of our Pi Alpha 293 AAV high-yield and quality production platform, which displays improved scalability and biosafety. Thirdly, we will also go over how we have been able to reduce the residual plasmid DNA that gets mispackaged into final AAV product by making some plasmid modifications. Fourthly, we will go over our strategy to reduce production costs by developing a new transfection reagent. And lastly, we will summarize what PackGene offers and how PackGene can be your one-stop DNA and AAV CDMO service provider. First, let's discuss the current challenges and the status of AAV-based gene therapy. A review written by Zhao et al. Identified 284 active clinical trials for viral vector-based in vivo gene therapies. They also noted that several vector modalities were used in active trials such as HSV, adenovirus, and AAV. The most predominant modality being used was AAV, accounting for 48.2% of the identified trials. This study also observed that clinical indications are expanding beyond rare diseases to broad-spectrum indications such as cancer, infectious diseases, and monogenic diseases such as ophthalmological, neurological, metabolic, and hematological diseases. If you add monogenic diseases together, you can see that monogenic diseases are the major indications of active AAV-based clinical trials. This goes to show that there has been a shift in the use of AAV for the treatment of more common diseases. The development of these life-saving gene therapies does not come without its challenges. Several companies face setbacks with their programs due to concerns regarding safety, efficacy, and chemistry, manufacturing, and controls. Some safety concerns involved adverse immune responses in patients and even patient deaths, as was the case for Astellas' Aspiral trial for the treatment of myotubular myopathy. Regarding efficacy, last year we saw how Sarepta's program for Duchenne muscular dystrophy had raised concerns regarding its reduced efficacy due to changes in manufacture, which had changed between the second and the third clinical trial. Finally, in June of this year, the U.S. Food and Drug Administration allowed the expanded use of Sarepta's therapeutic, now known as Elevidys. When it comes to CMC challenges, Pfizer, Freeline, and Orchard Therapeutics all face more stringent requirements from the FDA, such as assessing feasibility and the need to regulate maximal viral dosages. In the previous slide, I presented some of the safety, efficacy, and CMC challenges that AAV-based gene therapies are facing. But another challenge is seen at the point of manufacture, with GMP production facing scale-up and productivity gaps, especially for those therapeutics whose routes of administration require high dosages, such as intramuscular injection and IV administration. As an example in this table, you can see that Zolgensma is administered by IV and requires 1.1 e to the 14 VGs per kg. And in manufacturing, to achieve this viral titer, oftentimes requires between 45 to 200 liters of culture to generate a single dose. So in this example, you can see how challenging it is to produce the high viral vector titers needed for AAV-based gene therapy. There are other challenges, of course. One is the customer journey, where it is especially challenging to find patients for rare diseases that were previously untreatable. In terms of manufacturing, producing AAV vectors at a scale and quality sufficient for clinical and commercial use is challenging. The cost of goods remain high, partially due to the low and variable yields with limited manufacturing capacity. There are also clinical challenges. Navigating the regulatory landscape for gene therapies is complex, with stringent requirements for safety, efficacy, and manufacturing standards. Ethical considerations, especially concerning germline modifications and long-term effects, add to the challenges. Market access also presents a challenge. The high cost of developing, manufacturing, and administering AAV-based gene therapies can limit accessibility for patients, and ensuring that these therapies are affordable and accessible to a broader population is a significant challenge. Now we have a poll question for our audience. What is the current challenge you're facing with AAV therapy manufacturing? High cost? Productivity bottleneck? long turnaround time, finding a CDMO that can act as a one-stop solution. As mentioned earlier, the use of AAV has expanded beyond the treatment of rare diseases, and the fact that treatment requires high viral titers has led to an increase in the demand for AAV production. Greater than 2,000 preclinical and clinical gene therapy development programs exist. This increase in demand brings forth additional challenges, such as overcoming the long turnaround time. So currently, developers face long lead times for preclinical development, GMP manufacture. Another challenge is the high cost of AAV production. Development and production for a first-in-human viral vector program could cost around $5 million. The high demand also leads to a productivity bottleneck, where this demand exceeds current production capabilities by orders of magnitude. PackGene is positioned to address industry-wide productivity bottlenecks for AAV production while maintaining product safety and can serve as your one-stop shop solution.
Thank you, Tiffany and Bernice, for the introduction. My name is Ren Li. I'm the Upstream Process Development Associate Director here at PackGene. As Bernice mentioned, gene therapy has been facing various challenges, especially for the CMC part. In this section, I'm happy to introduce our Pi-alpha 293 AAV production platform, which was developed by PackGene, aiming for high-yield production of AAV vectors with improved quality. As indicated by its name, the Pi-alpha 293 system is an AAV production system based on in-house developed 293 cell lines. Considering different demands of our clients, we have developed both adherent and suspension clonal cell lines. The cell lines are generated by multiple rounds of single-cell cloning process. In the left picture, titers from the wild-type 293 cells are shown as gray bars, and the PackGene developed adherent cell line, also known as Pi293A, is shown in black. We can see for multiple projects, the PackGene cell line has outperformed the wild-type cells with increased titers up to 7-fold in certain cases. Similar to results of the adherent cell line, in the right picture, multiple in-house developed clones of suspension 293 cells, especially for those shown in blue, have demonstrated higher titers compared to the widely used VPC 2.0 cells from Thermo Fisher. The titer from clone 14, for example, is 4 times the yield of VPC 2.0. For clients who are interested in using those cell lines, we have generated the MCB and WCB for the adherent cell line. For the suspension cell line, we have already generated the master cell bank to support GMP manufacturing. As we know, other than the cell line, another component of any 293 transient transfection system is the plasmid. We have developed a proprietary rAAV cap plasmid, which is composed of wild-type rAAV and cap sequences, as well as a new non-coding element. By adding this novel sequence, the uniquely designed rAAV cap plasmid has been shown to improve AAV titers by 3 to 8 times in both adherent system, as shown in Figure B, as well as the suspension system in Figure C, comparing to the regular rAAV cap plasmids. Especially, this upstream yield increase has been observed in projects for multiple AAV serotypes, as shown in these two pictures. The proprietary cell line and plasmid I just discussed form the foundation of the Pi-alpha 293 production system, which usually gives improved titer from the start. For projects aiming for a high yield or the initial titer is not satisfactory, we can further improve the yield by optimizing the AAV upstream production process, especially for those critical process parameters for determining AAV yield. Considering interactions between different factors, we usually perform the optimization with a DOE-based approach. The optimization process has been focusing on 3 sections involved in AAV production, which are transfection conditions, upstream engineering conditions, as well as lysis and harvest conditions. Regarding transfection, for example, the critical parameters include cell density, total DNA amount, 3 plasmid ratios, and the ratio between DNA and the transfection reagent. After transfection, the stir speed, pH, DO, and temperature need to be carefully controlled in certain ranges to ensure optimized titer and quality. During the lysis and harvest stage, an optimized process can ensure the thorough release of AAV vectors, as well as efficient removal of host cellular DNA impurities without using too much expensive benzonase. The reduced benzonase usage while maintaining high-level removal of host cellular DNA impurities can lead to a significant reduction of production cost from both the reduced usage of the enzyme itself and a decrease in the size of filters used for downstream clarification. With those optimization procedures, we have observed a 10 times increase of AAV yield in certain projects. Our Pi-alpha production system not only has high yield, but also has demonstrated good scalability at different scales, as well as improved vector quality. The picture on the left shows the viral titers at harvest are maintained from 3 liter to 200 liter process. In the middle figure, from a 200 liter batch, we have achieved 1E17 total vectors at harvest. The AAV vector quality in this case, the residual whole cell DNA, as shown in the right chart, has shown a steady reduction when scaling up. We have our second pool question. What is the safety feature you value the most in AAV production? High full-empty ratio, low residual whole cell DNA and plasmid DNA, low level of whole cell protein, or low level of replication competent AAV, as known as rcAAV.
Using our Pi-alpha 293 platform, we have also put a lot of effort in developing and optimizing our process to improve biosafety by generating full capsid enriched material. While the most effective method of separating full capsids from empty capsids is by ultracentrifugation, this method at large scale proves difficult. Now companies are looking for alternatives to ultracentrifugation, and anion exchange chromatography has become one method by which empty capsids are effectively separated from full capsids. In this bar graph, we are showing percent full capsids for various AAV serotypes. The gray bar shows the full capsid percentage post-affinity, while the blue bar represents full capsid percentage post-anion exchange chromatography. The lowest enrichment we observed was with AAV6, and the highest enrichment was with AAV9. Overall, our optimized process results in full capsid enrichment ranging between 76 to 92% post-anion exchange chromatography. depending on the serotype that is being used. This percent enrichment is similar to results reported by other groups where their anion exchange optimized process led to a percent enrichment of viral capsids between 65 and 95%. By reducing key impurities with our optimized process, we have also observed improved biosafety and activity of AAV infection. Here, panels A to C are examples of the key impurities that we have been able to reduce. Panel A shows that by ultracentrifugation, we can achieve a high percentage of full capsids, greater than 98%. And with AX, we achieve a full capsid percentage greater than 90%. In panel B, we are showing the encapsulated plasmid DNA ratio in the blue bars, using our optimized process and a control process. As you can see, our optimized process contains 90% less encapsulated plasmid DNA than the control process. Panel C compares the residual host cell DNA between our optimized process versus a control process. The blue bars, again, show that our optimized process leads to a 95% reduction in residual host cell DNA when compared to the control. Panel D compares the biological function of an AAV vector carrying a luciferase reporter gene that was produced using either a traditional process versus PackGene's process. As you can see, the bioluminescence imaging obtained on day 14 illustrates that AAV vector produced using PackGene's process results in longer lasting biological activity. Overall, in this slide, we show how our process has achieved the reduction of key impurities, which in turn led to improved biological function.
As we know, one key process-related impurities is the residual plasmid DNA, especially the DNA that is encapsulated inside the vector and hard to remove by downstream process. Besides the pi-alpha 293 production system I just discussed, we here at PackGene have also developed novel plasmids aiming for reduced residual plasmid DNA packaging in the final product. With different plasmid designs, the left picture shows the viral titers are not affected by these designs. However, as shown in the middle and right pictures, the encapsulated plasmid DNA has been significantly reduced from about 3% to 0.1 or 0.2%. In this section, I'm going to discuss our strategy to bring down upstream manufacturing cost by screening new transfection reagents. It is known that gene therapy has very high production cost, and one big component of upstream manufacturing cost is the transfection reagent. For transfection of cells in 200-liter scale, some transfection agents can cost as high as $150,000 for a single batch. In the pie graph on the left, showing a cost analysis of a 500-liter batch, besides the plasmid, the transfection reagent shown in yellow contributes a big portion to the upstream cost. All those high-cost reagents and materials, combined with complicated upstream and downstream manufacturing processes, leading to a very high price tag on the AAV gene therapy drugs, as demonstrated in the table on the right, which lists some of the recently approved AAV drugs. Aiming to significantly reduce upstream manufacturing cost, we here at PackGene has been actively developing and screening new cost-effective transfection reagents. In Figure A, we have screened multiple transfection reagents, and the transfection reagent A has demonstrated a significant improvement of viral titer in the cell lysates as compared to the widely used Factovir AAV transfection reagent. The high titer with transfection reagent A correlates well with an increased transfection efficiency to over 80%, as shown in Figure B. After DOE optimization with this novel transfection reagent A, we have achieved an impressive upstream titer as high as 3.5 E12 Vg per mil in certain projects. With significant improvement on yield, the relative cost for AAV production have also been lowered. We look forward to seeing more applications of this novel transfection reagent A in our future client projects to make gene therapy more affordable.
Finally, in these last few slides, we would like to summarize who we are and why PackGene can be your one-stop plasmid, DNA, and AAV CDMO service provider. PackGene was founded in 2014 in Massachusetts, and since then we have become a leading AAV, CRO, and CDMO that provides reliable, economical, and scalable plasmid DNA and AAV viral vectors for early-stage drug discovery, preclinical development, and clinical programs for cell and gene therapy. PackGene offers over 110,000 square feet of GMP plasmid DNA and AAV manufacturing facilities. Currently, we are serving over 900 customers globally, from academic institutions, biotech, and big pharma companies. We have successfully delivered over 10,000 batches of research-grade AAV packaging service. In 2022, we began a construction of our new site located in Houston, Texas, which now has 35,000 square feet of GMP space that includes process and analytical labs, as well as 6 clean rooms, and even extra room that would allow us to grow for future expansion. This is the location that Ran and I are currently working at, and our process development and analytical labs are in full operation. The GMP production capabilities are expected to be fully operational during the second half of this year. Here we have presented how our Pi Alpha 293 high-yield AAV production platform has been optimized for improved productivity, scalability, and biosafety. We have shown an increased AAV yield with our uniquely designed rEAV plasmid. We have increased the total AAV yields by 10 times, up to 1E17 VGs in the 200-liter scale. We have methodically reduced the key impurities such as empty capsid, plasmid DNA, and whole cell DNA in the final AAV product. We have also showed how a new plasmid design results in reduction of encapsulated plasmid DNA by 90%. Overall, this platform includes 6 patents, and we have an additional 2 patents currently under review. Overall, we offer GMP plasmid DNA manufacturing, for which we have 2 GMP plasmid DNA production sites. Our production scale ranges from 5 liters up to 200 liter scale, with a production yield of up to 50 grams per batch and a lead time of less than 21 days. We also offer state-of-the-art 8,000 meters squared GMP AAV manufacturing. For this, we have 4 GMP AAV production suites, with production scales being between 25 liters to 500 liters. Our production yield can reach up to 1E17 VGs per batch. We offer production conditions for adherent or serum-free suspension conditions, and currently our lead times have no backlog. PackGene has a highly skilled team dedicated to delivering leading-edge methods for QC analysis of plasmid and AAV samples, with 50+ established analytical testing methods, including TCID50, replication-competent AAV, and analytical ultracentrifugation as examples. We offer analytical qualification and validation that is risk-based and phase-appropriate. Our analytical development QC platform covers greater than 95% AAV batch release and in-process testing. Since its founding in 2014, PackGene has established long-term cooperative relationships with 20+ multinational corporations and 200+ top institutions across Asia Pacific, China, Europe, and North America for a total of 28 countries. With the new US-based facility that is currently under construction, we will be able to serve our US customers better and, most importantly, be able to bring life-saving therapies to patients faster, more reliably, and more cost-effectively. With this, we are making great strides in advancing our mission of making gene therapy affordable. Thank you all for joining.
Thank you.
Thank you, Dr. Lee and Dr. Carol, of the insightful presentation. Sorry about the technical issue that we had at the beginning of this webinar. But right now, if you do have any questions, please put them in the Q&A chat box and we will answer them as we receive them. So just take your time, and we're going to start with the ones that already gotten during the presentation. So the first one is, what are the advantages and disadvantages of using transfection versus stable producer cell line system for upstream AAV production?
Yeah, thank you, Tiffany. The question is comparing the advantage versus disadvantage for upstream production of AAV vectors using either transient transfection or the stable cell line. Basically, there are three mainstream systems for upstream production of AAV vectors. The first one is triple-plasmid transfection of 293 cells. Second one is, as mentioned in the question, is using a stable producing cell line, PackGene cell line. Third one is using the SF9 baculovirus system. Due to the time limits, I think baculovirus system will be totally another topic. So we just focus on the transient versus stable producing cell line. For transient system, one of the biggest strengths is flexibility. If you have multiple gene of interest to test, or you have engineered capsid to compare, so transient system give you just most flexibility. Can simply switch the GUI or the capsid in your process, in the transfection process. However, for the stable produced cell line system, that will be hard to achieve. You have to develop a cell line to make this happen. The cell line development process usually involves screening hundreds of thousands clones, and usually takes about a half year, including the stability test. So that's definitely one of the advantage for transient systems. And associated with that, the transient system also provides us with the faster timeline to go to IND compared to the stable produced cell line systems. So, however, there's also a certain advantage for the stable cell line. First one is, I think you can once you define your cell clone by robust screening for a high yield and high quality, the process can be very robust and can be easily scaled up to a higher scale, like 1,000-liter bioreactor or even above. However, for transient systems, we know the transfection process It's a hard to control process. You face batch-to-batch variance, and you need a lot of upstream PD work if you decide to use a transient transfection system. And also the cost-wise transient transfection, you have to use GMP grade plasmid and the transfection reagent every single batch. So that's good, maybe good for initial project early phase. You don't involve so many patients. However, for late stage, stable produced cell line system will be easier. You don't use any of those expensive transfection reagents, right? So that's the biggest advantage for the stable produced cell line if you do multiple batches in late stage trial or you go to commercial. And generally, once the titer and the quality can be higher for stable produced cell line because you can screen the best clone out of your process, and the vector itself is generally more homogeneous. But for transient transfection, the vector can be heterogeneous and with different level of genomic integrity, DNA impurities. And another CMC risk of using transient transfection is if you use a novel transfection reagent, you also have to develop an assay to characterize those residual transfection reagents to make sure it's fully removed after manufacturing, because you have to report that to FDA during the IND filing. So thank you.
Thank you, Dr. Lee. Let's move on to the next question we got. So the next one is, what is the full empty ratio in your production?
So that's actually a great question. So we see different level of empty full ratios with different transfection region and also different cell line. So that's usually case by case. But for some of the lower end, we see about like for upstream, right harvest about 10 to 20% can go as high as 50% to 60% with different transfection reagents. But usually those numbers will change and also enriched during the downstream purification process. But generally we say about 10-20% to 50 to 60%, depending on the different transfection reagent and cell line we use.
Thank you. The next one we got is, how will you optimize the downstream purification process for the specific AAV vector?
Okay, so I can answer that question. So for us, we initially like to take a tailored approach. So I think everybody's aware that, you know, when you're working with different serotypes, there's serotype-specific differences as well as gene of interest. And so we like to take the time to really talk with our clients and try to get as much background information as we can so that we can develop our purification strategy. We like to be able to get some of the background, such as like your experience with any instability issues. If you have any buffers that you've seen that are not working in your program, then we can easily scratch them out from our downstream process development. And then the good thing about having such a large team here with us is that we have a lot of experienced people working with different serotypes. So then we can tailor yours and make small modifications if we've experienced or have worked with other AAV serotypes that you're working with. So we can leverage on that as well. But most importantly, I want to say that what we like to pride ourselves on is in having the tailored approach in trying to meet your expectations and trying to see what it is that you're trying to achieve with your vector.
Okay, thank you. Moving on to the next one. So what are the upstream challenges for scaling up AAV manufacturing process from laboratory scale to commercial scale?
Yes, thank you, Tiffany. So for upstream challenges, first one, I will say I can divide this challenge into two parts. One is the cost part. As I mentioned in the previous question, if we go to a higher level, you require a large quantity of GMP plasmid, and also the large quantity of transfection reagent. As 200 liter costs as high as 150K for a single batch. And also another big cost for upstream is the benzonase. So solution for that is we can choose a PackGene, already choose an alternative for benzonase and reduce the cost by 1 to 10%. So, and for the process part, the biggest challenge for scaling up upstream process, if we use triple-plasmid transient transfection, is the transfection itself. So it's involved optimization of the mixing method, right? So and selection of transfection reagent. And we know that based on literature, the optimal size for a PI-based transfection complex is 500 nanometer. So this 500 nanometer allows the cell can endocytose the transfection complex. Meantime, also let the complex get out of the endosome easily through proton sponge effect. So I think with different, there are a lot of ways to optimize to try to achieve that 500 nanometer if you use a PI reagent. Somebody try different adding a stabilizer, like a Prisma script. You can add a BSA to the transfection complex to elongate the incubation time to 1 hour before adding to the bioreactors. Or somebody designed some renal regenerative file. They play the different cell concentration in the transfection mix, try to also try to make this complex more stable and scalable before infusing to the bioreactors. Yeah, that's my
Okay. Yeah, thank you, Dr. Lee. So the next question we have on the QC. So for the QC, have you had any tests on the viral contamination or virus clearance on AAV product?
For the QC part, for the virus, I think since that's actually one thing I missed. If we use a stable cell line system, or I would say stable packaging cell line system, you will involve adding virus, a helper virus, either adenovirus or the HSV, to deliver the gene product to assist the gene replication in the 293 cells, or HeLa cells. But in our case, we use 293 based AAV transient transfection system, so there is no virus added to the system. However, there is still a chance to generate a replication competent AAV at the end of the process. So we already developed a very robust QC assay to characterize the rcAAV to ensure there's less than one rcAAV per E8 particles following the deadline.
I guess a follow-up question, what are some of the QCs that you guys offer for this platform?
Based on, I'm not from the QC team, but based on what I know, so I think PackGene can do most of QC release assay in-house. So okay. Yeah, pretty much we can do most.
Okay, thank you. The next one we have here, what are the biggest bottlenecks you have observed for downstream process development?
So I can answer that one. So I would say that there's several, at least the ones that are typically encountered or that I find are a little bit more challenging coming from clients are, for instance, scalability issues. Many times we have people coming from academia, so they bring over their laboratory scale process, and then trying to do a tech transfer from that is usually not an easy transfer. So I would say that that's a big one, because, coming from an academic background, the process is not consistent. You're using centrifugation or to clarify sometimes your lysate, whereas in manufacturing in a CDMO, you actually need to start introducing filter optimization and filter studies, right? So I would say that's one of the big bottlenecks that we have in trying to streamline a process that's coming from laboratory into a CDMO, for instance. Another challenge that I have found, or that people can always relate to, is a vector-specific challenge. So just because we've had clients where they come with one AAV vector, say for instance AAV1, and then for their next product they'll come with AAV9. So sometimes people think that the purification process is very similar because it's AAV, but it's not. It's actually quite different. The overall structure of the virus is different depending on the capsid that you have, right? The gene of interest also plays a big role. So it actually affects the charge of the particle. So even for
chromatography steps, such as anion exchange chromatography, it proves to be quite difficult to optimize a process, and that usually takes a lot of time. So that's another bottleneck where you might be stuck trying to develop a process or optimize a process just for one chromatography step. And I think the other bottleneck that we face is like the purity and yield. So a lot of that comes from the upstream, right? So if you have a very high yield, then it makes the purification process a little bit easier, just because you have a lot to work with and a lot of material to try to develop and finish a process. And so if your yield is low, that's usually a big bottleneck for us, because in downstream, in order to, for instance, take a product from affinity and try to optimize anion exchange, you need a certain amount of material to be able to screen your anion exchange. And if your yield is very low, then you can't even try to determine binding capacities or for your AEX step. So I would say those are the three major bottlenecks that people face: scalability, vector specific, and purity yield challenges.
Okay, thank you. I think that concludes today's webinar. If you do have more questions, feel free to reach out to us, and our speakers will be able to answer them afterwards. This webinar will be available on demand if you do want to visit back, if you missed out anything. And on behalf of PackGene, thank you for joining today's webinar, and we look forward to see you in our next one. Thank you. Bye-bye.
Thank you.
Thank you.